by hundreds of millions of variants that exist. And those variants, if you can characterize them, allow you to identify one individual's DNA from another individual's DNA with essentially almost infinitely reliable statistics. Also, another feature of DNA is that all of that inheritance stuff that we were just talking about, DNA is inherited in a Mendelian fashion. And so it means that if you find DNA at a crime scene of an individual who you don't know, by finding DNA of that person's relatives, you might be able to infer who the person at the crime scene is. So, for example, crimes have been solved quite recently by using 23andMe. 23andMe is a company, a commercial company in the US that does commercial DNA testing for people for fun. But that DNA has, what they do is they identify about 600,000 DNA variants in every person's DNA to do their studies and to produce their product. And it's possible to find DNA at a crime scene that you can't identify, but if you go into a database like 23andMe, there's a good chance that you could find that person's uncle's DNA present or their cousin's DNA. And you know that because of the inheritance patterns that we were just talking about. And so by looking at those inheritance patterns, you can infer whose DNA it is that you have at the crime scene. So this sort of Mendelian inheritance of the variation in DNA from parent to child allows a tremendous amount of power in identifying any particular DNA relative to a population of DNAs that are available. And so DNA, therefore, is a molecule that's useful in many ways for both identifying whose DNA it is and identifying which DNA you're particularly looking at. So when thinking about DNA variation, there are a number of forms of commonly used DNA variation that are used in forensic studies. The first of which was DNA that's referred to as variable length nucleotide tandem repeats, or VNTRs. And these VNTR sequences, we'll talk more about them in a minute, but basically they're relatively short sequences, somewhere between six and maybe as much as a few hundred base pairs in length, that are repeated tandemly in the genome. So at one position in a human's DNA, there'll be a sequence like you see here. Oops, I'm sorry. Like you see here, this GAATTCCATCC might be repeated 300 times in one person's genome. And it turns out that because of this repetitive nature of the DNA, when it's replicated by DNA polymerase, mistakes are made, and what happens sometimes is the DNA polymerase slips, if you will, on the sequence, and will change the number of copies present in that individual's DNA at that position. So one individual might have 100 copies at that position, whereas another person might have 120 copies of that DNA at that position. So the number of copies present at any particular place is a very important variable in everyone's DNA, and it's relatively consistent in every individual. So it makes it a very useful variation of DNA sequence that allows one person's DNA to be distinguished from another person's DNA. More recently, it's been identified that not only are these sequences available, but there are sequences that are shorter relatives of these VNTRs. And by shorter, I mean di, tri, tetra, up to about hexanucleotide length sequences that are found scattered throughout the genome that are repeated on the order of, say, 3 to 20 or so times at specific sites. And these are called microsatellites, as opposed to these VNTRs, which are often referred to as mini satellites because they're bigger. But the microsatellites are much more prevalent in the human genome. And in fact, about every 300 base pairs or so in the human genome, one can find a microsatellite sequence. And just like the VNTRs, these tandem arrays of genes, these are tandemly arrayed as well. And the length of these tandem arrays of repetitive sequences varies from one individual to another. So you can use the length of the microsatellite at a particular position as a variable genetic marker in that individual. And as I said, there are literally millions of these in everyone's genome. So it's easy to find these things and use them for this purpose. Finally, the other population of human variation that's used often, particularly in population genetic studies, and not so much in forensics because of the way the assays are done and so forth, but single nucleotide polymorphisms are present throughout all genomes, plants, humans, everyone. And single nucleotide polymorphisms are found about 1 every 100 base pairs in DNA. And they're the result of essentially random mutations that have occurred over the millions of years that any particular species has been on the planet. And in humans, we've only been on the planet as a species, as a kind of a breeding species by itself, for about 2 million years, which in evolutionary history is relatively a short time. But nevertheless, in that 200 million years, about 30 million of these single nucleotide polymorphic sequences have evolved in the genome. And it turns out, I'm not going to have time to talk about it, but that pretty much every one of those has been now identified on Earth, so that there are no new single nucleotide polymorphisms to be found as population markers in human populations anymore. They've all been discovered, which is a kind of a remarkable feat. That means that 30 million variants have been identified. And that information has been a very powerful tool in carrying out human genetic studies. But again, we're not going to deal with the single nucleotide polymorphisms today. But in your plant studies, you'll undoubtedly come across the use of SNPs in breeding strategies that you're going to be using in your plant work. And there are powerful tools now for looking at these SNPs in all organisms. And so it's worth, after today's class, it's worth going out and reading more about SNPs if you don't know what they are. Okay. So let's get back to the forensics question. So for the forensics stuff we're going to talk about VNTRs and microsatellites. And as I said, it has the features that they're inherited. You can use it to infer close relatives, and you could also use it to identify individuals unambiguously. And so just by way of keeping you, making sure you're still there, here are 2 DNA sequences shown at the top here, starting, this is one DNA sequence from one individual, and this is DNA sequence from a second individual. And there are changes. These 2 sequences are somewhat similar to each other. You can see that there's, they all start out G, G, A, G, T, G, etc. But there are variations that occur in this sequence. So can you identify which variations are present in these 2 sequences among the ones shown here? Microsatellite variation, mini satellite is the same as VNTR, SNPs, or combinations of those 2. So take a second and stare at the sequences, and then maybe, Ravinder, you could ask for a poll with A, B, C, D, and E.
So this time don't say, just think to yourself. You shout out. So that everybody gets a chance to take your confidence. Those who think it's A, raise your hand. 5, 6. Those who think it's B. A, B, C, D, E. So there are mostly E's, there are a couple A's, and there are a couple C's.
Okay, so somebody who picked E tell me where the microsatellites are.
So, from this to perfect T. So, would you like to come? So that he can also hear. YT. You are in the C? Okay. E. This is E. Don't feel shy. Nothing bad is going to happen.
So there are both microsatellite and SMG radiation filters. So at the fifth position, in the first sequence there is T, and in the another sequence there is A. So SMG radiation filter and mini microsatellites, that is nucleotide GT. So there is variable number. In second sequence there are more number of GTs in the second sequence. So microsatellite radiation.
Correct. Okay. So these sequences have both single nucleotide polymorphism here, T and A, and so one individual, these two individuals differ at this position, but they also have this repeating GT sequence of different lengths, right? So here the GT goes from here to here, whereas in this person it goes from here all the way to here, right? So there's an extra an extra 2 GT sequences in the bottom one compared to the top one. And so one could use those differences. If an individual with the top DNA was at the crime scene, you'd be able to distinguish it from the one at the bottom, right? And so that's the way this kind of variation could work in forensics. Okay, so the adoption then of using DNA as a forensic tool has been quite slow. And some of the reasons for that is, number one, the only way such evidence can be used in a legal proceeding is when the courts allow it. It's not something that the legislature can say you must use DNA evidence. It must evolve as a sort of a legal tool by courts allowing a prosecutor, say, to use it in a court of law. And it turns out that that process took about 30 years for it to actually come to fruition in the United States. And I think in some countries it's still not really the standard for identifying individuals. I don't know in India whether DNA evidence is widely used for forensic purposes. But anyway, the first use of VNTRs was in 1985 in Great Britain, actually, in a trial, and we're going to talk about that trial in a second. In that case, these VNTRs, to ascertain the VNTRs, was a major process that took months and months of analysis of the DNA found at the crime scene. And that was because it used a method called southern blotting, which is something that I think modern students don't even know what it is anymore, but basically it's a way to detect the length of these VNTRs using hybridization tools. One of the classic cases in the United States was the trial of a football player named OJ Simpson, who was accused of murdering his ex-wife. Was that trial heard about in India? Did people know about that, or is that just a U.S. phenomenon? They do know it, okay.
Just for 1 second. So OJ Simpson was a football player. So kind of think of that he is the Sachin Tendulkar or Virat Kohli of US. Yeah? But he was that famous there and did something bad. Yeah, go ahead.
Okay. So anyway, it was that trial captured the American press for over a year because they used DNA evidence, and the trial was actually presented on TV, kind of live, and so there was a lot of dramatic courtroom scenes on the news in the U.S. On TV, basically every night for many months. And so with VNTR, besides the fact that it takes a long time to do these southern blots and hybridizations for DNA, it takes a lot of DNA material to be recovered at the crime scene, which is to say that you have to prepare enough DNA to be able to detect it using this so-called southern blotting technique, which requires literally microgram amounts of DNA, which isn't a lot of DNA by standards if you're preparing DNA. But if you're trying to recover DNA, say, from a blood spot in somebody's shirt, getting a microgram of DNA from that is very difficult. So anyway, so it needs a lot of DNA. Then as technology advanced in DNA sort of sequencing work and in the laboratory, that technology moved into the forensic labs. And so people moved from using these VNTRs to microsatellites, which, like, for example, CA repeats, because these assays were possible to carry out using the polymerase chain reaction, or PCR, which allows you to use vanishingly small amounts of DNA. In fact, you could do PCR, as probably you know, on a single molecule of DNA, and you could still detect the sequence that you're interested in. So this use of microsatellites instead of VNTRs evolved gradually, but is now the method of choice for doing forensic DNA studies. It can also be automated so that commercial laboratories can carry out hundreds or thousands of assays a day without too much difficulty, and the accuracy of the assays is tremendously reliable. And so, and then over time, other newer methods using the polymerase chain reaction combined with fluorescent methods and automated machines have moved the time scale from literally months to carry out the analysis, like in the OJ Simpson trial, to really DNA evidence can be had in a matter of a couple of hours from isolation of the DNA to the results. Single nucleotide polymorphisms can be used for this purpose because there are a lot of them, but the technology for doing that in crime scenes is still a little problematic, and so it's not likely to be used anytime soon for forensic studies. Okay, so typically when you're at a crime scene and you're isolating DNA, or if you're in the field and you want to isolate DNA from plants to look at inheritance patterns or whatever, DNA can be used from a number of categories. The first is genomic DNA, which we've been talking about. That's the entire genome, basically, of DNA. But also in all eukaryotic organisms and plants contain mitochondrial DNA. And mitochondrial DNA is a much simpler DNA, so it can be used for some purposes. And in addition, at least in humans and in mammals, there's sex differences are determined by XY inheritance patterns. So there's a sort of a bi-chromosomal mechanism for determining sex. In plants, it's a lot more complex. So Y chromosome analysis obviously doesn't really apply to plants. Genomic DNA, some of the advantages are tremendous amounts of variation in the form of SNPs, microsatellites, VNTRs. Insertions and deletions are common in all human DNA. DNA is readily detected using PCR methods. The human genome has been fully sequenced now for about 20 years, and so we know a lot about what to expect from the DNA when we characterize it. So it's got that advantage. And as I said before, inheritance can be inferred. So even if you don't know the identity of the DNA you're studying, you might be able to figure out who the individual is using public databases to determine it. There was a famous paper in Science magazine in the US about 5 years ago where people took SNP DNA and took SNP information from an unknown source of DNA, and within 10 minutes they were able to identify whose DNA it was by the pattern of SNPs using only public DNA databases, so using things like 23andMe and other publicly utilized databases, and it was done by doing this inheritance pattern with cousins and uncles and aunts. So, you can't hide in plain sight anymore if your DNA can be found. Some of the disadvantages are, DNA, as you heard from Ravinder, is subject to DNA modifications, methylations, and so forth, and that can sometimes affect the analysis of the DNA sequence. Sometimes it's hard to get intact DNA, depending on the nature of the source material. So, for example, anthropological samples that are very old have damaged DNA. And sometimes crime scenes where there are fires or other sorts of things, sometimes isolating intact DNA is difficult. And then PCR amplification might be sometimes dependent on the nature of the source of the DNA. Mitochondrial DNA has some advantages, which is to say that mitochondrion have small genomes, typically on the order of a few tens of thousands of nucleotides instead of the billions of nucleotides present in human DNA, so limited amount of variation, which can be an advantage because it makes it simpler to find the differences. It has SNPs and it has insertions and deletions that can be used. It's very easy to purify and obtain because mitochondrial DNA is usually circular. Circular DNA is protected from degradation by the environment. And so, particularly in anthropological samples, the first studies were all done in mitochondrial DNA because that was the only DNA that could be isolated in sufficient quantity. That's since been overcome, and genomic DNA has been used too. It's fully sequenced in humans anyway, and in many other species, so using it is quite easy to do. However, mitochondria only come from the mother. So you can only tell if an individual at the crime scene has the same mother as you think they might, but it's possible that if you can't rely on the maternal inheritance, then it can be problematic. Okay. Y chromosome DNA is similar problems. It's only useful really in paternity studies because it's passed only in the male germ line. And particularly if the criminal happens to be a female, you're out of luck. Okay, so molecular methods then for forensics that are widely used include actual DNA sequence analysis, which is not currently the method of choice, but one day will become the method of choice as sequencing technology gets cheaper and quicker and more accurate. VNTRs and RFFPs can be analyzed using very crude methods like Southern blots and other ways, but because the VNTRs can be quite long, they're not often used well. They don't work well with PCR. Other methods to look at polymorphisms include some methods that are specific to companies. This one by a company called ABI, involving a fluorescent assay and PCR. Other companies use these so-called gene chips, so Affymetrix and Illumina have these arrays, and these technologies are very useful for SNPs, not so useful for microsatellites or VNTRs. And then there are really high-tech methods that use mass spectrometry that allow you to determine some of these sequence polymorphisms very, very rapidly and accurately. The only limitation here is the cost. So it's often expensive to use mass spec compared to other methods. Okay. So how do you use these DNAs to study actually crime scene DNA? So the sort of forensic DNA sequence analysis traces its origins back to a fellow that's called Sir Alec Jeffreys. Alec Jeffreys was a regular scientist studying genetics in Leicester, in Great Britain. He was a biochemist by training, but got interested in DNA and repetitive DNA sequences. He moved to Leicester after being basically a postdoc at Oxford for a while, and he started to look at variation of sequences of these VNTR sequences. And so here's Sir Alec explaining his discovery, for which he received a lot of accolades, and he became knighted as a result of it. And I think I fixed the sound, so let me know if the sound is legible. If it's not, we'll just skip it, and I'll tell you what he said.
When people refer to restriction fragment length polymorphisms, also known as RFLPs, or SNPs, single nucleotide polymorphisms, these are just different techniques for measuring DNA variation, which all of us have in the very long 3 billion base DNA sequence. So they're the DNA bases that make up the genetic code, the genes that cause the proteins, and variations both within the genes and between the genes can be used to follow patterns in families. Another use of them that many people are aware of is for DNA fingerprinting, identifying within a blood sample a specific pattern of variation which is characteristic of an individual. A very well-known example of this was the OJ Simpson trial, where the DNA fingerprinting, I think, or RFLP technology was discussed a lot in the popular press.
We discovered this shared sequence motif shared by these different mini-satellite repeats. So we thought, okay, what we need to do now, if we're going to detect lots of these really good mini-satellites, is you take this shared motif, make it as a tandem repeat, and use that as a probe. That should be very good at detecting mini-satellites. So I think it was mid-September, did the experiment, I think it was on a Friday, left the X-ray film exposing, and the result came off on Monday morning. I think it was about 9 o'clock in the morning. But I just went into the dark room, we'd done this experiment on what we call a blot, just a little membrane containing various DNA samples. It was pretty messy. It was very, very messy, but it was clearly working. What we're detecting were lots of bits of DNA, and in the humans at least, they're clearly variable, very variable. In fact, what we got was our first very, very murky DNA fingerprint. And at that point, the penny dropped. I thought, wait a minute, we've actually solved a totally different problem here. In theory, if these patterns, if we can improve this technology, these patterns are going to look as though they're going to be extremely individual specific. And not only that, we can begin to see from our little family group, but they seem to be rather simply inherited as well. So as if a child was made up of half of mum's bands and half of dad's bands on this in the DNA fingerprint. So I thought, okay, we've got possibility here of using this for identification. So we immediately thought forensics. Possibility here, maybe we could use this for establishing family relationships. So immediately started thinking of paternity disputes. We also thought about other potential applications, and these are all thoughts that were coming in a very embryonic form into my mind, literally within the first 5 minutes of actually peering at a bit of X-ray film.
Okay, so it's interesting, I think, always to hear people like Alec Jeffreys describe the thought process that he went through to to make a discovery that's basically changed the world in many ways, right? Because it was that blot that he's holding up there in front of his face that you see in the picture in front of you. That kind of image immediately made him realize that DNA is very variable and that each individual has a characteristic set of bands, and you could use those bands to identify different individuals, different parentage, different where they came from and where they're going. Okay, and so just to put a finer point on it, what basically you could show using those patterns that the pattern of bands is inherited in a completely strictly Mendelian fashion, so that if you can see that shown here, it's a southern blot basically with somebody who's homozygous for this band, somebody who's homozygous for that band. These are VNTRs, that is the same sequence location in the genome, but this one has more repeats than this one. And this individual is basically the offspring of these two and has inherited one copy from the mother and one copy from the father. And so they look heterozygous. So you actually see these patterns, in these kinds of gels. You could actually follow these patterns if you know who the individuals are. You could actually follow the pattern of the NTR bands. So from parents, you could infer offspring, and you can infer which chromosome came from which parent. As I said, he was knighted for the discovery of that, and that discovery then was put into rapid use, because that discovery was actually made in the sort of 1980s, mid-1980s, and the first case that it was used in was for this guy here named Colin Pitchfork. And so basically, there was a series of 2 women were murdered and raped and murdered in a small town in England. This person named Richard Berkman came forward. He was 17 years old and confessed to the first murder and rape and denied that he did the second one. However, his blood type, which was then, in those days, was a common way of identifying a person in a rape case, where they used the blood type ascertained from the semen recovered from the victim, actually matched Richard Berkman. And so basically, he confessed, his blood type matched, and he was thrown in jail. But it turned out that that was also the time when Sir Alec had discovered his DNTR-based fingerprinting, and so he carried out DNA analysis on the semen that was recovered from the victims, from the 2 murdered women. And it turned out that the fingerprinting made it absolutely impossible that Richard Berkman was the guy who did the crime. And in fact, he was just confessing because he was somewhat, he was a little bit dotty, he was a little crazy, and just felt like he wanted to be found guilty. So what the police then did was, once they realized that he wasn't the guy, the police spent the next 6 months collecting blood and saliva samples from all of the men that lived in the same town that these 2 young girls who were murdered came from. And it turned out there weren't a lot of men. There was only about 5,500 of them in this town. And they did the DNA fingerprinting on all of them, and not a single one matched the DNA in the semen of these guys. So it was possible that the person came from another town, or that they missed something, okay? And so it turned out that while this investigation was ongoing, a guy in town who was just a guy who likes hanging out at a bar, named Ian Kelly, overheard that this guy, Colin Pitchfork, got this guy, Richard Buckland, to provide his blood sample for him and claim that his name was Colin Pitchfork. So the DNA, so Colin Pitchfork never actually gave a sample of his own DNA. And they never had it in this original 5,500 men that they rounded up and obtained DNA from. But after the police overheard that, they convinced Pitchfork to give up his blood for a screen, and it turned out that it was a match. And so Pitchfork was then convicted of both murders. So this occurred in the late 1980s, and there are at least 2 movies that I'm aware of that actually are not documentaries, but they are dramatizations of this event and the process of collecting the DNAs and so forth. So this points out a number of things. Number one, it points out how powerful DNTR-based DNA fingerprinting is, because it's unambiguous, and it's particularly unambiguous in only 5,500 men, because there are millions and millions of possible combinations of DAMs, and in this small number of men, the likelihood of anyone sharing exactly the same pattern with the semen is vanishingly small. But the other thing that this points out is that there's still police work to be done, right? So if you're using DNA evidence, it's very important to know the source of the DNA and how the DNA was ascertained, and infer information about where the DNA came from in the first place. And without that information, some of the best DNA sequence information is not going to be useful, right? And so over time, those processes for collecting the DNA and analyzing it have become very carefully controlled. Okay, so the next case is the so-called OJ Simpson case, which again, as Ravinder told you, OJ Simpson was one of the most famous football players in America. And he was a real personality. He was on TV all the time. He was, on talk shows, and he was a star. He starred in many movies. One day, his ex-wife was found murdered in her home in Hollywood, California. And so at the murder scene, it was a very bloody murder scene where the wife was murdered using a knife. And so there was blood everywhere, all kinds of blood all over the place.
Can I pause you for 1 second?
Sure.
So on this day, when this murder happened, I was a postdoc and I came to the lab. So I was sharing my bench with somebody else. This somebody else was a fan of football and he was a fan of OJ Simpson. So when I came to the lab, so he said, have you heard what happened to OJ? So to me at the time, OJ meant orange juice. I just went from New Delhi to there. I had no idea about, if he said something about Sunil Gavaskar, I might have meant something. Right? So anyway, he thought that I'm going to be showing some response. I was like, Dad has cucumber. I said, yeah, what happened to orange juice? All right, go on.
Okay, so at the crime scene then, there was blood everywhere, as I said, and so they collected the blood and did this VNTR analysis on the blood, and then they did it on all of the people who were at the scene, including the dead woman, whose name was Nicole Brown, shown here on the right. They got DNA from this guy, OJ Simpson. He was a suspect right away because he was the ex-husband. So there was a suspicion that he could have been the killer. There's a guy named Ron Goldman who actually rented an apartment in the backyard of this gigantic mansion where Nicole Brown lived. And then there are a bunch of other people at the scene of the crime and some control people here. And then shown in these lanes are, this was blood that was obtained from the gate of the house. It was smeared on the side of the house when, obviously, when the killer fled the scene. This is a boot print that was blood on the floor where the killing occurred. More blood from the floor of the foyer, the room in which the body was actually found, and so forth. And so these are some of the examples of the DNA. And shown here are the hybridization patterns of the VNTRs. Okay. And so it was discovered then by looking at the blood that didn't match Nicole Brown, the victim, her blood, and you can see here's a band, and so forth. This DNA, there was blood found that was not her DNA. It was not Ron Goldman's DNA, because this was his pattern of band, but it was this DNA. See, there's 3 bands here and 3 bands here. 3 bands here, 3 bands here, 3 bands here, and 3 bands here. And so there was a very strong concordance then between the bands present in OJ Simpson, one of the prime suspect, and blood found on the floor and on the walls, and it was not the victim's blood, and it was not some other suspects, and they tested the blood of many people actually. It wasn't just this group. And so it was, you could see that it matches perfectly between OJ Simpson and these things, right? And so it seemed like an open and shut case, right? So there again, you can see these bands. I think you don't have to be a forensics expert to see that they're identical, right? And so, but in the United States, criminal trials have this requirement that the person has to be found guilty beyond a reasonable doubt. And a reasonable doubt is something that the law defines as what a reasonable person might think is an alternative explanation, right? So a reasonable doubt means it's a very high standard. It means that the prosecution has to prove the case to a very, very high level of certainty. And so when OJ Simpson ended up hiring some of the most famous lawyers in America to defend him in this crime. And one of them was a guy named Alan Jeffreys, who you may have heard of. He's a guy who just testified in the impeachment trial of President Trump in the U.S. And defended President Trump as well. So Alan Dershowitz has a history of defending bad guys. Excuse me, that's a political statement. But anyway, so he was part of the trial. Sorry? Yeah, I shouldn't say those things. Okay, anyway, so reasonable doubt. And so OJ Simpson was found not guilty by the jury because there was reasonable doubt that this data was correct. And it wasn't so much that the bands didn't match and so forth, but it was based on other things like the blood sample that was used by the police department to produce these bands disappeared for a day somehow. It was basically in the crime scene room, but they couldn't trace it for every second of its existence. And so the defense said that somebody actually was trying to set up O.J. Simpson for the murder, and that he had nothing to do with it, but they put his blood into the crime scene blood just to confuse everyone. Also, the company that actually carried out this analysis was a company called Solmark, which in its early days had made 2 errors in its VNTR typing. And so they brought that up repeatedly, even though it was 6 years before, two errors. And so after those pieces of evidence were presented, the jurors decided that O.J. Simpson didn't do it. And among the other things were that the jurors were completely baffled by the scientific evidence. They didn't understand a thing about it. And so when the evidence was presented in court, scientists came up, scientists like me, tried to explain what these results were. And as you can see, I'm not very good at explaining anything. And so and these guys weren't either. This was a comment from one of the jurors after they acquitted O.J. Simpson. I didn't understand the DNA stuff at all. To me, it was just a waste of time. It was way out there and carried absolutely no weight with me whatsoever, with me at all. This was one of the jurors. And that's the problem with DNA evidence, is it's highly technical. And so if you're not a technical person and you're trying to decide the guilt or innocence of somebody, maybe even to be executed for the crime, you want to understand the evidence. And so it took many years. This trial happened in 1995. It took many years for people to figure out how to explain DNA evidence to a jury of non-scientists. So if you're ever asked to serve on a jury of a criminal case, you might end up being the person who has to explain it to your fellow jurors. So always keep that in mind. It's always important to be able to explain your science in terms that non-scientists can understand. Anyway, so he got off on this. Sorry?
Let me ask them a question. So is there a jury in India when there are these legal court cases? Pardon? There used to be. There's no more jury. So there's no jury in India.
Really? So who makes the decision? And just the judge?
CC, just the judge. Yeah. And I can say that from watching all the Bollywood movies, that every time things happen, they go crossfire back and forth, and after that the judge comes and says the court is recessed, we'll meet tomorrow, and tomorrow he comes back with something and he says guilty or not guilty, and the life is done. And as judge, I'll be very.
I think that system sounds like it could be corrupt.
And I'll be strange here that in fact, when the witnesses come, they can be all intimidated. That whatever they want to say, they may not have the guts to say. And sometimes you can just give them $10 and you can make him say or her say whatever you want him to say for just $10.
Yeah.
Or even less. Okay, just a cup of coffee is good enough. And
okay.
So there's a problem here.
Anyway, yeah. The point here is that DNA evidence may be irrefutable, like it is in this picture, but it's not always enough. But I will say that over the last now 25 years that DNA evidence has become used, it has now evolved from being something that I didn't understand this stuff at all to basically everybody in America believes that DNA is foolproof, and that, you know, any DNA evidence is actually the only standard they believe in. So it's kind of evolved by just sticking with it and being persistent. So, you know, another recent use of DNA sort of evidence was that you're probably all aware, on September 11th in the U.S., the World Trade Center in New York was destroyed, and thousands of people were killed. And you can see in the rubble here, there were many bodies that were buried in this rubble that were completely shredded. That just small fragments of the people remain. And yet their loved ones, the family members, wanted to be sure that the person actually died at the World Trade Center. And so the government took on the task of filtering through all of this rubble and looking for pieces of humans. And they took those pieces of humans that were in the rubble and they subjected them to DNA fingerprinting, and then they used it to match it against the family members of the people who said their loved ones were missing, to identify a little more than half of the people who were killed in the World Trade Center. So it's a very powerful tool, and it shows that the durability of DNA as well, because, you know, this was very harsh conditions, and yet the DNA was sufficiently stable to be identified. Okay. So, I'm going to finish up in a few minutes. I guess you guys are going to break for lunch. Is that the idea in a few minutes? I'm sure I'm not certain of the time. Anyway.
I think they need tea before the lunch.
Yeah. Okay. So we'll break. I'll be done in 5 more minutes. Yeah. So is that okay?
Yeah. So.
Okay. Yeah. Okay. So this shows, again, in this picture, it shows a picture of mini satellites, and here you can see the mini satellites are shown in color. So this unit in the green is a mini satellite, and then this unit in the gray is exactly the same sequence as that. And here it's repeated 1, 2, 3, 4, 5, 6, 7, about 25 times, right? And so this is what amounts to a VNTR. And I assume you all know what PCR is. One could use PCR by using this fragment here, this fragment in red, as one primer, and this fragment in red as the other primer, to amplify a piece of DNA that includes this mini satellite. And then the length of the fragment that's produced from this PCR product is a reflection of the number of repeats present between this primer and this primer. And so you could use that now to use mini satellites or microsatellites to determine the number of copies present in a DNA sample at a particular position. So this shows it in schematic form, where in the OJ Simpson trial they used restriction fragments, and they used this hybridization with unique DNA to identify the length of the fragment from R to R. And so this is the fragment from R to R, and you know, each one of these has a restrictions fragment for pico-R1 at the end of it, and in between are variable length numbers of these repeats. Simple tandem repeats, or these microsatellites, as I said, are shorter. Here, these are microsatellites that are 5 base pairs in length, and again, it shows the variation that can occur. So this is one person who's a heterozygote. He has 2 copies on one chromosome, and he has 4 copies of the repeat on the other chromosome. A PCR primer that's located in this blue DNA, and out here in this blue DNA, would amplify a piece of DNA, and the length from here to here is a reflection of the number of copies of this microsatellite present in the DNA. So this individual would be a heterozygote with 2 repeats plus 4 repeats. Individual 2 would have 3 repeats on this chromosome and 3 repeats on this chromosome, and so this individual would be a homozygote. Individual 3, again, is a heterozygote with 4 on one chromosome and 6 on the other. So these simple tandem repeats can be used very effectively to look for polymorphisms that are present in a particular place. Again, the same primers are used in every person because they prime in the unique DNA that's outside the repetitive DNA, and the length of the repeats, the number of repeats, in this case 32, in this case 28, so forth, will determine the length of the entire fragment. And so when you separate the fragments by size, by acrylamide gels, or now more modernly using automated machines like capillary electrophoresis machines or DNA sequencing machines, you can measure the lengths of these repeats quite effectively. Okay, so based on that, in the U.S., the Federal Bureau of Investigation, or the FBI, you've probably all heard of the FBI anyway, is a federal organization that solves crimes in America. And the FBI, about 15 years ago, developed this so-called panel of microsatellites called CODIS, which stands for Combined DNA Index System, or CODIS, where they identified 20 alleles, 20 different microsatellites that are present at unique locations throughout the human genome, most of them on different chromosomes. They use these same microsatellites in every individual in which they do DNA sequencing and DNA typing. So every person who's basically accused is convicted of a felony in America now must give up a DNA sample, and this CODIS panel is performed on it. And each locus produces a number of alleles. So, this locus, there are 14 alleles. This locus, there are 30 different alleles. That means sequences of different lengths present in those individuals and so forth. And this panel, then, you could store each person's fingerprint of genes in a computer. So you would have 20 unique sizes for each person. And stored in a computer. So now when you find a new piece of DNA at a crime scene, if the person's CODIS DNA is on file with the FBI, you can identify that person in a few milliseconds by just putting in the CODIS sort of fingerprint from the crime scene. And so there have been efforts in some states to collect that DNA from large numbers of people. In the U.S., however, privacy is a very important factor, and individuals who have not committed crimes don't want their DNA on file. They feel like it's an invasion of privacy, and so most states have laws that prohibit the collection of DNA from anyone who hasn't committed a crime. The FBI has argued that if they can get this information from everybody at birth, they could identify everybody in the United States within a few seconds by their DNA. And it would allow the solving of many, many crimes at a very high rate. And so, one thing you might think about over lunch is whether if you lived in a country like this where the FBI has the ability to do this, whether you would want your DNA on file. And in my classes here at Colorado, we sometimes have a whole class devoted to this because people have very differing opinions about it. Many people have the attitude that if you've never committed a crime, what do you have to hide? There's no reason not to give your DNA. And others say that it's none of the government's business what the crime is. So anyway, something to think about. And I'll just give you one example of one more crime that was solved in Great Britain using DNA, and it was also a very high-profile crime. And if you have access to the BBC, this show called Crime Watch on December 21st is actually online. You could see it, and they go through this in great detail. But there was a creepy murder of this person named Mabel Leishern, who was murdered, and she was murdered in a way that made it seem like a vampire actually killed her. The person collected her blood in a saucepan and drank it, and other things. And so, and the crime was committed in this town that's in the newspaper is spelled that way, but the actual town this is in Wales, which is outside of England, it's in Great Britain. That's the way the town is actually spelled. So it almost looks like a polymorphic DNA sequence. And the pronunciation of that is quite remarkable here.
Conveyor Puckinge, Gogerish Mindrobo, Hantisilio Gogogo.
And that's the name of the town. So you should be thankful if you live in Delhi. Much easier to pronounce. For this crime, investigations show that there was no supernatural element in the crime. They found footprints in the glass under the broken door. There were bloodstains on it, and they hope to use that evidence to solve the crime. And in this case, they were able to isolate DNA from tiny amounts of blood. I'm sorry, I'll go through this very quickly. And in this case, they used the CODIS panel to do the identification. And so this shows how the CODIS panel actually works. These are 4 markers from the CODIS panel. Each one of these is a different microsatellite that is used. And shown in this pattern is what the pattern of microsatellite bands would look like using capillary electrophoresis. The orange bands show there's molecular weight markers, and this shows all of the alleles for each of these markers. So just as a quick question, if you're looking at a single allele, THO1, how many alleles of THO1 can any one individual possess? Quickly. A, B, C, or D?
B. Those who think it's 2, raise your hand. Those who think other than 2, raise your hand. Mostly thinks 2.
Yeah, okay. That's correct. That's good. You're paying attention. So, no person can have more than 2 alleles unless they happen to have like a trisomy or something like that. Anyway, so this was the actual evidence that was revealed. So again, this is the pattern of all of the available markers. At the crime scene, this is the pattern of bands for this marker with these 2 bands. For this marker, THO1, or this band and this band. For this marker, this band, and so forth. So this is the evidence that was at the crime scene. This was the evidence of Mabel, the victim. Right? And so clearly different than this person. And so these are two other suspects. So which one did it? 1 or 2?
One or 2. Those who think it's 1, raise your hand. Those who think it's 2, raise your hand. 1.
Correct. Right. And that's because you could see that this band, these 2 bands match these 2 bands match these 2, whereas this guy is homozygote for that marker, and this guy is heterozygous for that marker, and so forth. And this kind of analysis is exactly the way the police actually do it. So there's nothing different about it. That's what the actual data would actually look like. So it's quite impressive and quite accurate. Okay, and so at the trial, it's often questioned whether the DNA evidence alone has enough power, if you will, to show that that the victim had to, that the guy who's accused had to have done it, and that nobody else could possibly have that same pattern of bands. And so, for example, shown here is the allele frequencies for the alleles present at the THO1 locus. And so there's one with 5 repeats, 6 repeats, 7 repeats, 8 repeats, and so on. And this is the population frequency of those alleles. So it's known that 1 in 200 individuals in the population will have that allele, 1 in 4 will have that allele, etc. So the question is, if you found a person with 5 and 7 repeats, what's the probability in advance, without knowing anything else, what probability would you expect to find a person with those 2 alleles in the general population?
So the thing is B.
B?
So hold on. The problem with the shouting is that other people didn't get a chance. Other people will just, those who didn't get the B, They are now with B. Anyway, they think it's B. Or
I
C.
think it's B. So how did you come to B? I think it's important to hear how you made that
Or you
calculation.
think it's A? Raise your hand. B. C.
B. C. Okay.
Couple of hands up on C. D. Lot of hands up on D. E. D. Mostly.
So what's the answer mostly?
D.
D. Yeah, so the answer is D, and the frequency is the so-called multiplicative rule. When two independent events occur, the probability of the two events occurring is the multiplicative product of the probability of each independent event. And the frequency of having the 5 allele is completely independent of the frequency of having the 7 allele. And so the actual combined frequency of being a 5-7 individual would be the frequency of 5 times the frequency of 7, which is 1200. Okay. And so that's for one marker. And so then you do the same calculation for the entire 20 marker set, and this number quickly becomes 1 in trillions. Okay, so the likelihood of having all 20 markers matched at a particular, at 20 different loci becomes vanishingly small, smaller than the entire population of Earth. And so it means that this methodology is incredibly precise, and the only way errors that sort of misidentifying somebody could occur is in the case of identical twins, for example, or something like that, where they would have exactly the same allele frequency. So the police work identified this guy. They found DNA on a blood in his pocket, and so forth. And so he was found guilty of murder and sentenced to life in prison. So just to summarize that, before we break, the FBI has a database now of about 15 million individual DNA profiles. And so, as I told you, given 15 million, that's interesting, but also using the information from these 15 million and knowing the millions and millions of other people who are not curated in this database but exist in places like 23andMe and public data, it's possible using the public data plus this data to identify people with much higher numbers than just 15 million. So it's not just 15 million people who can be identified, but there are now in America probably tens of millions, at least, people who could be identified readily by their DNA. And again, this is something I hope you'll talk about at lunch, about whether you think it's a good thing, whether the government should have the ability to identify you by your DNA if you've done nothing wrong in the first place. Anyway, it started out with 350,000 investigations, but it's also exonerated 350 innocent individuals who had served a combined time of 4,700 years in prison, and now they were, and some of them were on death row, and they were exonerated by the fact that after DNA testing became available, it was determined that their DNA was not at the crime scene. And so it allowed those people to be released from prison. It's identified many more than 149 now. That was several years ago. And it's been involved in the conviction of many, many individuals now of sexual assault and other things. And so there are actually programs now in the U.S. where there's a project called the Innocence Project, which we're going to talk about after lunch, that involves the testing of all prisoners on death row, prisoners who are scheduled to be executed by the state for crimes they committed, and they're trying to get all of them tested and released from death row. And so far that program is incredibly successful. It's exonerated several hundred people now who would have otherwise been put to death. So it's a very important methodology to understand. Okay, so, and with that, I will, why don't you go have some food, come back and I have some papers that I gave. I don't know if you have copies of them, Ravinder. If you have a chance to make copies. If not, we could just do it on the screen. And you could solve some crimes and predict some frequencies and experiment with using some of these SPR methods for identifying individuals in populations. So, see you, in a half hour? Yeah, I just want to say, be gentle with me here. It's approaching midnight here in America, so and I'm old, so staying up late is a problem. But anyway, I'll let Ravinder decide.
So, what is your preference? So one possibility is that we'll hand out the sheets that you have sent it to me, and they can do those in the afternoon, and we can let you go.
You can do that, and I can send you, if you want, by email. I could send you like answer keys. Okay. So you could go through it. It's quite straightforward.
Yeah. Okay. So maybe that will be the best thing. That you send me the answer keys.
Okay.
We will let them do it, and then we will the answer keys to go through, and then there are a couple of things that we are trying to wrap up here. That way, you are not waiting for us, and they are able to finish, a few things, including this piece, in a more flexible manner.
Sounds good. Okay, so after the class leaves, I'll send you the stuff.
But maybe if you can just use a few minutes to kind of give an overview that what we expect from those exercises.
Yeah. So the sheets that you will get have on them some sort of descriptive information about using DNA profiles to identify people. And then you're going to go through and identify repeats and count repeats and make predictions about what crime scene DNA will look like, and then in the end determine whether the person did it or not, basically. So you're going to be looking at data like the data I just showed you, only you'll be given a little less information. You'll have to do some calculations and so forth to come up with the probabilities of your identifications and things like that. Make sense?
And yeah, so that's wonderful. So before he goes, so I hope you agree that he's a hardcore geneticist, and he has lots of wonderful things. Of course, not about the field that you are interested in, but the point is that you can learn from lots of things from what they have done, and hopefully use them for something that are of interest to you. Right? That's the whole point of so-called cross-fertilization between the different fields. So on that note, I hope, if you agree with me, that a wonderful presentation, let's just give him a big round of applause.
And you too. Thank you for listening, and for being patient.
And that's a compliment, so thank you so much.
Okay.
All right, so we will let you go to sleep. And I should also mention that he was away for a week. He took the flight today, and he just spent the whole day traveling, and now he's up here, and I think he deserves the sleep. Even though he has agreed that he'll be awake till 3 or 4 o'clock, because I won't be able to answer the question for his practicals. But I think we'll let him go, and we'll deal with that, and whatever comes out, we'll figure out a way to do one more study. Yeah? Okay. All right, my friend. Again, thank you so much.
Okay.
Yeah.
Thank you.
Yeah. Bye.
Okay.
Good night. So the plan is. So the plan is that just to get the most out in this concluding session, so we'll have our tea, and what time is the lunch? 2 o'clock. Huh? 2 o'clock. So I thought lunch used to be 12 to 1 o'clock. That's no longer true. All right, so 2 o'clock.